So normally when we do this lab, we break it up into 2 days. The second day is spent with the gel electrophoresis. But today I'm going to do the gel electrophoresis for you. And so what I want to show you is how you would actually be doing that if you were in the lab. So I have my setup here. I'm going to put my camera down so you can see. So what I have down here is I have a gel tray, and this gel tray has these things called rubber dams on them. So like a dam that stops up water, one on each side. And then we have this thing called a comb, and the comb looks like a comb. But what it does is these teeth are going to make little grooves in the gel substance. Now what we're going to do is create a nice gel, so a solid surface through which we're going to run our DNA. Now this is the gel box. In the gel box, eventually we're going to put our gel in there and we're going to run it through that, and that's the electrophoresis part, because it's got the little electric leads, and we're going to plug it into a high-powered, high-voltage electric machine. Before we do that, we're going to pour our actual gel. So this is some agarose that I've made already. I took it downstairs and microwaved it. It's pretty warm because it was boiling, and it's made with agarose, and it's in 0.8% gel. And that just means I took 8 grams and put it in 100 milliliters of what we call TAE buffer, and that just means tris acetate EDTA. So that's just a kind of buffer that's going to help neutralize charges as the DNA runs through the matrix. Now what I usually do is I start pouring on the side opposite the comb, and so I'm hoping that this is enough agarose to go all the way across. It is, yay! And so you keep pouring until you've basically filled up the tray, and that's all it is. Now right now it's liquidy, but as time elapses you will see it start to turn cloudy as it becomes solid, as it cools down to room temperature and becomes solid. So I'm just going to let you stare at that for a little while I go check on our samples. All right, so the gel is obviously solid by now. You can kind of see on the side that it's pretty solidified. And so when we take it out, we want to be really careful not to break the gel. And so with these rubber dams, it's actually really easy to do that. So we want to, sometimes I actually take a pipette tip and try to break the seal along the edges. That helps sometimes break it off a little bit easier, like that. We need to do the same thing on this side to break the seal. Pop it off. Nope, didn't do it all the way. I broke it a little bit, but that's okay. All right, so now we're going to put it into the gel box, and the way that it goes in the gel box, there's a little notch that fits, and when you have it perfectly down, the comb just pops out. And then we're going to put some electrophoresis buffer in. And what this does is it gives it the overall, nope, I broke it. It gives us the overall charge, so that the current that goes through the gel can be nice and even and go all the way across. So it's a little bit hard to see, but I actually did break the combs off, but I still have 3 decent wells that we'll use to actually load. So let's gonna be a little bit tricky. I'm gonna have to redo my camera angle so you can actually see what I'm doing here. So let me get that set. Okay, so I have pulled our samples out, and what I am going to do next is add a little bit of dye. So this is a loading dye, gel loading dye, and I'm going to add that to our samples for a couple reasons. One reason is it makes the sample a little bit heavier so it can fall into the wells, and the other reason is that it kind of lets you see where you're putting the sample. So it helps you visualize the sample as you're loading it onto the gel. So I'm just going to do that real quick. Add that to a couple samples since we don't actually get to run all of them since I broke the gel. So it looks like we have 3 samples. So I'm going to prep 3 samples. You can hear my pipetter clicking as I change my tips out. Make sure I don't contaminate anything. And there we go. Okay, so I have my 3 samples. And I'm going to keep them in the same order. So what I'm going to do is I'm going to load as much as will fit into the wells. So I'm actually going to set my pipetter at 10 microliters because that's the most that this particular pipetter will hold. Now I'm going to get as much of my sample as I can, and then I'm going to position my pipette tip so it is actually over the well. And I like to kind of use my other hand to brace it. So I'm going to have my pipette tip nice and centered over the well, and I'm going to gently kind of layer the sample in the well by kind of going back and forth. And I don't know if you can see this on the camera. It's kind of a light blue color that shows me where the sample was put. Now if I do that with the next sample, so we're only going to have room for 3 samples because of the gel issue. I'm going to do that again. I'm going to slowly, I'm not going inside the well, I'm just over the top of the well. And you can kind of, hopefully you can kind of see that I'm just layering it into the well and it's settling down on its own. Now some of that sample actually I just saw it pop out. So if you have a bubble in the well, it can kind of keep it from going down, depending on how well you actually cast your gel. So I'm going to do the 3rd sample here. I'm going to get that loaded. And it's a good idea to, you know, obviously take your time loading, but you also want to be pretty steady and timely while you're loading as well. You don't want it to be just sitting in the gel box too long because it can eventually kind of start to dry out and pop up above the wells. At this point, we're going to be running the samples through the gel using a current. So for the current, we need to use these this lid with the leads. It's going to, oops, I didn't mean to do that. It's going to actually pop on top, and it shows you, it actually lets you know that you match the black side to the black side and the red side to the red side, and that ensures that the current is going in the right direction. And that's important because you want the positive side, the positive charge to be on this end and the negative charge to be on that end, because DNA is negatively charged, so we want it to run in the direction of the positive charge. Now, we're going to take these leads and we're going to plug them into a power source. And this power source is also color coded. So hopefully those of you who can see color can do this. So we have a black one and a red one. And you notice that I used the same pair over here. So now I'm going to turn it on, if I can find the switch. And we are going to run this for a, it actually takes a pretty good amount of time. And so we need to set it to a voltage that is going to allow our samples to come into the gel at a nice nice pace. So they come into the gel and they get kind of stuck there, it's trapped there. So we want that to be around 80 is good, that's good. So this is the voltage we want to run. We've set it, and now we have to tell the machine that we want it to run. And so you'll watch it kind of come up to its voltage that we set. Now an indication that it's working, you actually can't see it from this angle, but you can actually see the bubbles going. See the bubbles? That tells us that the voltage is going through the wires and that the current is good. And so if you see those bubbles on each side, you know that the wires on each side are good. And I see the bubbles on both sides. This is a platinum wire that's used in these gel boxes to make sure that it doesn't degrade, being used constantly over and over in the gel buffer solution. Ooh, that cord definitely wants to be up there. All right, so I'm not gonna make you sit here and watch this because it's gonna take about an hour to do this. So the next bit of the video, you're going to, or even if I just present it in class, you're going to get a picture of the results from this gel or from another gel like that. I ran another gel earlier so you could actually see the results on the same day. So you'll be able to see the results and analyze the results once they are done. So this was your introduction to electrophoresis, and hopefully next year or next fall, or if you're around anytime in the spring, we can kind of go over it together and you can see how it's done in person rather than with these terrible videos that I do not know how to publish and polish to make them look good. So sorry about that. I'm learning just like you guys are. All right, so this is electrophoresis.